Welcome to the Ph.d. defense of Ása Jacobsen. Ása Jacobsen has been enrolled as a Ph.d. student at the University of the Faroe Islands. Her advisor at the university has been Professor Svein Ole Michaelsen. She has done the main part of her research at Fiskeelling, the Faroese Research Station of Aquaculture, where she has had Jonard Esterskær as a local advisor. With us on Zoom, we also have the evaluation committee, Sissel Beate Rønning and Philip Pope from Norway, and Samuel Martin from Scotland. Welcome. And the Ph.d. defense will proceed as follows. First, Ása will give a presentation of her work for approximately 30 minutes. Then the opponents will have up to 2 hours to ask questions. Afterwards, there will be a possibility for the audience to ask questions ex auditorio. Due to this somewhat special form of the defense, questions need either to have been sent to me prior to the defense or has to be posted in the Q&A forum in the Zoom webinar for this meeting. Then there will be a short break where the committee will go offline to assess the defense, and afterwards they will return with their conclusion. First, I'll give the word to Ása for a presentation.
Thank you. Welcome to the presentation and defense of my doctoral thesis with the title Exploring Potential Causes of Gaping in Salmon fillets. First, a short outline of the presentation. I will first introduce the problematic issue of gaping and some context, followed by the purpose and objectives of this work. Since I will talk about the experiments and analysis performed and the results. And finally, some thoughts on relevance and applications. So Atlantic salmon is a well-established aquaculture species. It is the sole species currently farmed in the Faroe Islands for food production and export. It is also a major aquaculture species in Norway, Chile, Scotland, and elsewhere, as you can see on the map. The production has increased dramatically since the 1980s, as can be seen on the graph in the lower right corner. The yearly production amount of salmon can be seen as the dark blue region on the graph, and the light blue region represents marine finfish species in general. This increase in production is also increasing the demands for sustainable production. One sustainability issue is low fillet quality, which causes problems in the processing and further along the supply chain. Gaping is one of the quality problems that needs to be dealt with. Gaping is the phenomenon where the connective tissue, mucomata, fails to hold together the adjacent muscle sheets, causing holes and tears in the fillet. The two images on the right side illustrate a fillet with intact tissue and a fillet with gaping. Gaping affects the profitability of the industry because of the difficulties in secondary processing into more specialized food products, and also because it loses the appeal to the consumers. It also affects the effort to increase sustainability due to the loss and wastage that it causes in processing and further along the supply line. Therefore, it is important to improve the quality and enhance optimal utilization of the salmon fillets. Now, gaping has been studied for many years, and research and efforts of improvement in the industry have led to optimizations. However, causes and mechanisms of gaping are still largely unknown. Previous research has had widespread focus, as can be seen on the illustration in the right corner. It seems not to be heritable, and for example, seasonal variation, various diets, collagen content in the tissue and such, often give inconsistent results when analyzed and do not provide any fulfilling explanations. Stress-induced gaping is the most widely accepted explanation, which has led to optimizations, although there is not always consistent results, and complete understanding of the process is still lacking. The purpose of this study was to gain insight into potential causes of gaping in salmon fillets and possible molecular mechanisms of this process. The research objectives were to first establish any potential effect of two production parameters on the occurrence of gaping in salmon. The tested parameters were cleaning of the abdominal cavity and temperature between slaughter and processing. And second, to determine if analysis of glycosaminoglycan content and structure in the connective tissue in the fillets with and without gaping can reveal potential mechanisms of this degradation. And third, to determine if bacterial enzymes potentially play a role in this post-mortem degradation in the connective tissue. And finally, to estimate the applicability of the used methods for research in this area. The analysis of the production parameter has been published in the scientific journal Aquaculture Research. We set up an experiment at a salmon slaughter facility belonging to a local farming company, Hiddenfjord. 30 salmon were used for examining the effect of post-slaughter temperature and cleaning of the abdominal cavity after gutting. Prior to slaughter, all salmon had experienced the same circumstances from fry to slaughter, and standard procedures ensured reduced stress levels throughout leading up to the slaughter. The fish were killed by a blow to the head and gills cut, both procedures performed automatically. And at this time, 15 salmon were put in storage boxes at ambient temperature. The other 15 salmon were put in a container with iced seawater and transported to the processing plant while chilled. At the processing plant, 15 of these 30 salmon were cleaned meticulously well, free of blood, kidney, and intestinal remains, while the others were only gutted and rinsed lightly. All 30 salmon were since stored in storage boxes with chipped ice in a refrigerator container. All 30 salmon were examined following a standardized quality estimate procedure, including 27 parameters. Some measured on day 1, before storage, and others after 7 days. Some of the measurements are illustrated in the photographs to the right, and the sensoric evaluations are listed in the table. Methods of examinations were established through implementation of standard procedures such as QIM, and staff training in a previous project on salmon quality led by Magnus Pauli Magnussen. On day 1, the temperature between slaughter and processing was measured by temperature loggers, while the sensoric evaluation of the cleaning of the abdominal cavity was performed by trained staff working with these examinations at a regular basis. The staff was unaware of the different treatments, and the cleaning of the abdominal cavity was graded 0 to 3 using the guidelines listed below the table. On day 7, the salmon were taken out for quality measurements, again performed by trained staff. Fillet gaping was measured by lifting the fillet carefully and sliding the hands down along the length of the fillet. This exposes potential holes and slits, as seen in the photograph. The number and sizes of these tears determines the grades assigned. A multivariate analysis was performed, including all samples and variables in the experiment. I will mention only a few here. The manipulated variables, temperature and cleaning of the abdominal cavity, are seen in the top left corner and to the right of the figure. Other measured variables are presented with letters, and variables centered around origo without relevance for the plot were removed for clarity. The symbols represent the individual fish, and the temperature and cleaning of the abdominal cavity can be seen to have the largest influence on the positioning of the other variables and the samples, as they have the largest distance from the origo. Gaping is illustrated by the letter A and is drawn towards the cleaning variable, and the multivariate analysis therefore suggested a correlation between cleaning of the abdominal cavity and gaping. This was supported by a biplot comparing only these variables. No correlation was detected between gaping and the temperature between slaughter and processing. However, this might have been due to the relatively warm temperature of the supposedly chilled salmon group, which was caused by a failure in the cooling system at the time of the experiment. When comparing the salmon that were cleaned well and those who were not cleaned, a significant difference was detected in gaping scores assigned to the two groups. As can be seen in the figure, the well-cleaned salmon, illustrated by the gray bars, were assigned gaping scores 1, 2, and 3, while the not cleaned salmon, illustrated by the black bars, were assigned all but 1 to gaping score 3. This finding indicated that something in the blood, or intestinal fluids, or remains induced or affected the severity of gaping in salmon fillets. In order to analyze the connective tissue more closely, samples were taken from each of the 30 salmon in the experiment described earlier. Following the quality measurements, samples of connective tissue were taken as indicated by the black boxes on the pictures on the left. Each salmon provided either one intact sample colored in green, or one gaping sample colored in red, or one intact and one gaping sample as colored in purple. In total, there were 43 samples, and the results of the connective tissue analysis were published in the scientific journal ACS Omega. Here in the upper right corner, there's an illustration of the connective tissue between the muscle layers in the salmon fillet. The myochomata is an extracellular matrix composed of different molecules interacting with each other and the muscle cells. It provides structural support, adhesiveness, and means of biochemical communication within the tissue. The center of the myochomata is named an interstitial matrix and is mainly composed of fibrillar collagens, such as collagen type 1, which is illustrated in the lower left corner. Proteoglycans with attached glucosaminoglycans are illustrated on the collagen here as red strings, and they help organize and develop the collagen structure and maintain its integrity. The interstitial matrix determines physical characteristics such as physical strength and elasticity. The basement membrane, now illustrated on the left, lies between the interstitial matrix and cell surface of the muscle cells. It contains other collagens and proteoglycans and links the cell to the extracellular matrix, and together providing attachment and possibility of interaction between the cells and its microenvironment. The basement membrane is dominated by collagen type 4, which forms two-dimensional networks and are again also highly reliant on proteoglycans and glucosaminoglycans for structural integrity. The basement membrane is of particular importance as the site of disruption when gaping occurs seems to be in the basement membrane and on the muscle cell surface. Now, the proteoglycans consist of a core protein carrying glucosaminoglycans. The main type of glucosaminoglycans are illustrated here on the right. They are all linear carbohydrate polymers composed of repeating units of disaccharides. An amino sugar illustrated by the squares, and a uronic acid or galactose illustrated by diamonds or circles. Most types are usually modified with sulfate groups, illustrated by the small red circles. The various types of glucosaminoglycans vary in their composition, but within each type there's also a variation in chain length, chain structure, sulfation degree, and pattern. Now, because the glucosaminoglycans are relatively large and highly charged, they can dominate the chemical properties of the proteoglycans, and therefore the type and number of glucosaminoglycans attached to the proteoglycans, as well as their characteristics, is of great importance for the function of the proteoglycans. Chondroitin sulfate and heparan sulfate were selected for analysis as they were expected to be most abundant in the tissue analyzed. Here is the chemical structure of the disaccharide units of chondroitin sulfate and heparan sulfate. The potential site of sulfate group modifications are illustrated in red. Chondroitin sulfate is commonly sulfated on one or both of the potential positions on the amino sugar, while sulfation of the glucuronic acid is not as common. Heparan sulfate is a more versatile compound as it displays more possibilities in disaccharide composition, and it also has a larger variation in sulfation pattern. The 43 tissue samples taken from the salmon fillets were frozen, pulverized, and lyophilized prior to the extraction of all glucosaminoglycans. Following extraction, part of each sample was digested with APC chondroitinase and part with heparinase 1, 2, and 3. Due to lab restrictions or instrument time, the number of samples had to be reduced prior to mass spectrometry analysis. Therefore, some samples were pooled, but each pooled sample consisted of no more than 2 individuals, and pooled samples were either both gaping or both intact, and they were from salmon getting the same treatment and getting the same gaping score assigned. This resulted in 32 heparan sulfate samples and 32 chondroitin sulfate samples. Here is a figure with representative extracted MS spectrum and chromatograms for the analyzed chondroitin sulfate. Subfigure C shows a representative extracted MS spectrum covering the retention time from 80 minutes to 130 minutes. Various peaks are labeled with MSAT values and ion species description. The small table on the left describes how the ion species are named. For example, we can see the small peak on the lower left in the MS spectrum is labeled 00111, which means it is composed of 1 amino sugar, 1 sulfate group, and 1 acetyl group. The highest peak in the MS spectrum is therefore composed of 1 unsaturated and 1 saturated hexuronic acid, 1 amino sugar, 1 sulfate group, and 1 acetyl group. Now, all peaks known to represent chondroitin sulfate compounds were analyzed by making extracted ion chromatograms as illustrated in the subfigures D, E, and F on the right. And these were used for quantification. This procedure was performed for all samples of chondroitin sulfate and heparan sulfate. From the mass spectrometry analysis, it was possible to analyze and estimate total amounts, sulfation degree, chain length, chain structure, etc. Looking closer at the results for the chondroitin sulfate analysis, The aim was to digest all the chondroitin sulfate disaccharide units, but larger oligosaccharides often remain in low amounts, so they were included as well. The table gives an overview of all oligosaccharides included in the analysis. Focusing first on the disaccharide, they're in the red box illustrated on the table. They were divided into groups with 0, 1, or 2 sulfate groups as illustrated by the red circles. Comparison of these types were made between intact and gaping samples because the sulfation is important for the functional characteristics of the glucosaminoglycans and hence the proteoglycans. The other oligosaccharides were also compared and included when estimating chain length. As illustrated earlier, we got one intact sample from some of the salmon, which were green, one gaping sample from some, which were red, and one intact and one gaping sample from some, which are purple. Comparisons and statistical analysis were made between intact and gaping sample using all samples, as illustrated in the upper right corner. Also, we used only those samples which were from individuals where both one intact and one gaping sample was extracted, as illustrated in the lower right corner. When comparing all intact and all gaping samples, there was no significant differences detected using the Welch's t-test, as you can see on the table on the far left. No, right, sorry. The comparison included total disaccharide amount of the 3 disaccharide types, and the various oligosaccharides. Also, the estimated chain length and the relative content of the 3 disaccharide types was compared. However, when comparisons were made between samples where intact and gaping samples were taken from the same individuals, there was a significant difference in the amount of the monosulfated disaccharides and also in the distribution of the non-sulfated, monosulfated, and desulfated disaccharides. The paired t-test was used for statistical analysis, and the Wilcoxon test gave the same results. The figure in the lower left corner shows a dot plot with the relative content of first non-sulfated disaccharides, then monosulfated, and then desulfated disaccharides. The strings connect the samples taken from the same individuals. As you can see, the gaping samples have a higher relative content of non-sulfated disaccharides and a lower relative content of both mono and desulfated disaccharides. Since the gaping tissue had a lower sulfation degree, while there was no difference in the total amount or chain length, it suggested that the gaping tissue had been subjected to more desulfation, but not degradation. One possible explanation for this is the action of endosulfatases. Although the endogenous chondroitin sulfate catabolism has not been fully established, the most recognized path of catabolism, degradation of the chondroitin sulfate in vertebrates, is by fragmentation into oligosaccharides, followed by sequential degradation from the non-reducing end. To our knowledge, no eukaryotic endosulfatases have been reported. However, bacterial endosulfatases have been detected in the marine environment and human intestines. It is therefore a possibility, though not a certainty, that the detected differences could be due to bacterial activity. In order to investigate the microbial community present in the post-slaughter environment of the salmon, as well as the potential gelatinase activity, we made an experimental setup. We were gracefully allowed by Bakkafrost Farming to utilize their facilities and salmon for the experiment. And we started by taking whole intestines from 15 salmon straight after slaughter and manual gutting. These were put in sterile bags and frozen immediately for sampling at a later time, where biological material from 2 to 3 salmon were pulled into each of the 6 samples from the esophagus, the stomach, pyloric ceca, mid-intestine, and distal intestine. From the automated processing line, we extracted another 17 salmon just before the final rinsing stage. We rinsed 9 of them manually and put them into 3 storage boxes with chipped ice, and put the other 8 into 3 other storage boxes without rinsing them. These boxes were since stored in a cooling facility where we sampled skin mucus, abdominal cavity, mucus and fluids, and storage ice the following 7 days. This experiment with analysis and results has been published in the scientific journal Ph.d. And some quick technical notes. The samples were homogenized, and part of each sample was used for DNA extraction using various QIAGEN kits corresponding to sample type. The library preparation for sequencing was prepared largely according to an Illumina library prep manual, using the recommended amplicon primers from Clintworth et al., covering the V4 and V4, 3 and 4 regions. An initial test was performed using a few samples for each type for estimating proper concentration to use for the amplicon PCR, as this could be affected by host DNA and other things. And also to include negative controls in the various DNA extraction kits, for example. This test resulted in minor adjustment, though the major change was to move from the initially tried polymerase to using kappa high fidelity polymerase, which gave better results. When we were happy with the setup, the analysis were performed for all samples. The community composition detected in the storage ice can be seen in the figure to the left. The 4 bars on the left represent the storage ice pulled from storage boxes where the salmon was rinsed before being stored. 1 bar for each of the sampling days, 2, 3, 4, and 7. The 4 bars on the right represent the storage ice from the other boxes. Again, 1 bar for each sampling day. And one key element here is the trend of a development over time on the microbial composition. This is especially apparent in the storage boxes where the salmon was not rinsed, the bars on the right. The microbial community in the skin mucus did not show the same trend of a change over time. And in the pie chart to the right, each pie represents one of the storage boxes with sampling days pooled. The bacterial composition in the skin mucus seemed to reflect a mixture of microbiomes. Some bacteria known from the marine environment and similar to reported from skin mucus, and also some that probably were from the digestive system, such as Mycoplasmataceae. And this was also more apparent in the storage boxes where the salmon had not been rinsed. And also some that might be from storage ice or the water used during processing. None of the samples from the esophagus were successfully sequenced, and only 1 of the 6 stomach samples was successful. The other 3 sampling areas in the digestive tract were more successful. The pie chart on the left represent the average bacterial composition in the samples from stomach, pyloric ceca, mid-intestine, and distal intestine. As you can see, they are highly dominated by 1 or 2 otius. Mycoplasmataceae is seen as the light blue, and Alivibrio, which was also relatively abundant, is illustrated in dark blue. In contrast to this, the bacterial community in the samples from the abdominal cavity shown in the figure to the right was more diverse. Each group of 4 bars represent 4 sampling days in each storage box. Clearly something was different in storage box 3, which was highly dominated by Mycoplasmataceae. And when comparing the content of Mycoplasmataceae in the various boxes using the ANOVA-Tukey test, storage box number 3 was significantly different from the other boxes. However, when comparing this with the digestive tract samples, there was no difference. This indicated that there had been a transfer of fluids from the digestive tract which was not cleaned away during processing. A multivariate analysis supported this finding. It is shown here in a 2D plot with PC1 as the x-axis representing 34% of the variation, and PC2 as the y-axis representing 22% of the variation. The circles represent the various samples, storage ice in yellow, digestive tract in green, abdominal cavity in red, and skin mucus samples in purple. The letters represent the OTUs with influence on the positioning of the samples, as those close around the origo were removed for clarification. The position of the intestinal samples in green is clearly influenced by parameters A and B, which represent Mycoplasmataceae and Allevibrio, which also were highly abundant in those samples. The red circles closely related to the intestinal samples represent the abdominal samples from storage box 3. The storage ice samples were influenced by other parameters and did not correlate with the intestinal samples. Gelatinase activity was measured in the same samples used for sequencing analysis. Detection of general gelatinase activity was measured by using the NCHEC Gelatinase collagenase assay kit. The substrate gelatin in the assay can be degraded by several enzymes, including gelatinases such as MMP2 and MMP9. Therefore, this is not a measurement of a specific enzyme, but rather the collective gelatin degrading activity. An initial screening of a few samples of each sample type, shown in the graph in the center, showed that the gelatinase activity in the skin mucus and abdominal samples was very low or near absent, while the pyloric ceca and mid-intestine had a very high gelatin degrading enzyme activity. The distal intestine had also a relatively high activity. The figure to the right shows the results from all abdominal samples analyzed, and the black bars represent the samples from storage box 3, and the gray small bars represent the average of the other storage boxes. There was clearly a high gelatinase activity in the samples from storage box 3, which is consistent with a transfer of intestinal fluids and bacteria from the intestines to the abdominal cavity, as seen earlier, and the demonstrated high gelatinase activity in the intestinal samples. An ANOVA test showed a significant difference between these samples and the other abdominal cavity samples. Though the results indicate that the enzyme activity originates from the intestines, it does not say anything about the origin. If the enzymes are from the salmon's blood or intestinal fluids, or if it is of bacterial origin or a mixture thereof. Now, if one wants to look closer at the potential of bacterial influence and applying microbiome analysis, there are some challenges that one should keep in mind. There are many sources of potential contamination during sampling and while working in the lab. These can skew the results. Also, the bioinformatic analysis of the data contains many options, and these can also have an effect on the results. In an initial pilot study we performed prior to the analysis just presented before, using other samples, we saw the same dominating presence of Mycoplasmataceae in the salmon intestines presented in the first 3 bars in red. In another fish species, link, which is presented with the green bars to the far right, we saw a relatively high content of Mycoplasma, a genus in the family Mycoplasmataceae. Although this was a very limited study using different primers and methods, it suggested that these were closely related but not the same bacteria, and which could be distinguished in the reference database. On the other hand, when analyzing the same raw data from the published microbiome study presented earlier, but using a different bioinformatics pipeline, including a different reference database, we saw that the reads originally identified as Mycoplasmataceae, the blue large bubbles on the top, were now identified as Mycoplasma, shown in orange. This just illustrates that the results can be influenced by the library preparation and bioinformatics pipeline chosen. For analysis of actions of specific bacteria, it is therefore good to have supporting or complementary methods. And to summarize, the quality estimations show the importance of cleaning the abdominal cavity properly, which is relevant and applicable for the industry, the sustainability effort, and for future experiments. The mass spectrometry analysis of glycosaminoglycans demonstrated the good potential of the method to clarify molecular differences of interest. It showed differences in the chondroitin sulfates and suggested endosulfatase activity. This can be relevant for future studies. The microbiome analysis gave a good insight into the processing of salmon and revealed relevant bacteria. These results are of importance for the industry and for the future studies. And finally, thank you to all collaborators working on this with me. Thank you.
Yeah, we thank Ása for her presentation. Now the committee will have up to 2 hours to question Ása. The committee consists of Ph.d Verja. She is a scientist at the Norwegian Institute for Food, Fisheries and Aquaculture Research, NOFIMA, and she is the chair of the committee, and of Philip Pope, who is an associate professor at Norwegian University of Life Sciences, and Samuel Martin, who is a professor at University of Aberdeen. And Philip and Samuel will be the 2 opponents asking questions, and I hereby give the word to them. Here you are.
Hello. Thank you very much. Can you hear me okay now? Yep. Okay. Thank you very much, Ása, for a very interesting overview on some details there about the studies and the background of your PhD. So just to let you know what Philip and I have planned is I will concentrate initially on the introduction and your first 2 results papers. Philip will probably add on to the discussions as he feels appropriate when he's got some questions he wants to interject, and I will maybe occasionally ask Philip if he has anything to add to some of the discussion areas. And then Philip will concentrate very much more on the latter 3 papers on your microbiota analysis. Ása, if you have any issues with understanding me, just ask me to repeat a question in case I'm speaking too much or too fast. So I'd just like to, I'm really upset that I was not able to visit the Faroe Islands, because I would love to get a trip there, but that's the way life is at the moment. So I was just going to ask you some general questions about the aquaculture industry in the Faroes, just to get us going, so I can have a little bit of background. So maybe could you just start off by telling us a little bit about the size of the salmon industry in the Faroes and how it is organised?
Yes. There are currently 3 farming companies. One is Mowi, which was formerly called the Norwegian one. You know that one, probably. Yeah. It does not have that many sites here on the Faroe Islands. And then there is Hidden Fjord Farming Company, which also is relatively small, but focuses much on quality, mainly, high quality. And then there is Bakkafrost, which has a much larger part of the industry and is also more involved. It's involved in Scotland now, I think, as well. Has bought some, in there. So they are the 3 sort of parts in the industry currently.
And I found a very nice little video about Faroese salmon farming on the internet a few days ago, and they were saying at the moment that the Faroese use 0 antibiotics in their production. Is that still the case?
Yes, it has been so for quite a while. It had a great collapse in the, early 90s, I think. Can't remember exactly when they had major outbreak of disease and such. And after that, they implemented some very strict rules as to transport between areas and rules for sort of regulating these issues a lot better. And that has resulted in a very good organized and using low degrees of medications and such.
Yeah, so it certainly seems like we're going for a premium product, premium market. So just one more question really about the salmon farming in the Faroes. If we compare that to Norway and Scotland, does the Faroes have any unique challenges which maybe we would not see in the other European farming countries? Is there anything particular about the Faroese situation which maybe makes it distinct?
I think the main problem is getting enough area for it. We're small and it's exposed, so using the fjords is very important, but they are all taken at some point, and then you have to move further out, and that is highly challenging.
Yeah. So the things I was thinking about was probably more high energy sites. Something else I noticed when I go to Scotland, the west coast of Scotland, is that we have a lot of Eastern European workers on the fish farms, a lot of Polish, Romanian, Slovakians.
Okay.
And I was wondering, in the Faroes, are you mostly local workforce, or do you also have a lot of sort of international workers as well?
Oh, it's mainly locals, I think.
Okay, that's good. Yeah. Anyway, so those are just some little background questions I was interested to ask you. So your PhD is really trying to look at this downgrading of fillets, and particularly understanding how the caping affects the salmon product. You didn't expand onto other sources of downgrading of salmon fillets. So, can you think of any other problems associated with fillet quality, but also has an impact on the sort of quality?
Well, the soft fillet is quite often discussed, and it is shown here that it is also affected by this cleaning of the abdominal cavity, but it doesn't seem to be the same. There can be soft fillets without gaping, and the other way around. So that is one issue. Then there are also issues of dark spots, and so there are plenty of them, I think.
Yeah, so in my notes I had pigmentation, which is exactly what you're talking about, melanisation of some of the flesh.
Yeah.
And also what we see in Scotland, I believe in Norway as well, as adhesions as well, possibly following vaccination.
Okay.
Where you actually get adhesions, and that again can downgrade the fillets.
Okay.
Yeah. Separate to gaping, but these are other quality issues that I know the fish farmers are trying to avoid as much as possible. So you referenced, if we go now into more into the Ph.d, those are sort of some of the general questions, but you mentioned that up to 38% of the fish could be affected by gaping by a paper published by Karen Pittman in 2013. Mm-hmm. That's now, I know you've been doing this PhD for a few years, but I was just wondering what the current number might be. Has there been improvements in production protocols that might maybe have reduced that since those papers were published?
Yeah, I think it was also shown in the paper there that it varies a great deal from facility to facility. So this 38% is probably not the norm. I would expect it to be a lot lower. And it has also been improved, certainly. But there seems to be sort of in some time spans it seems to be a problem again. So and I think it's mainly because they don't really know exactly what affects it and what exactly is happening, and then some of for example, they have been working a lot with the stress factor and the cleaning factor as well, since I mentioned it, and that has improved quite a lot, I think. And I mentioned somewhere the project that was made earlier at Fiskalink, led by Magnus Paule Magnussen. That was where he established all these quality estimates and such. He did a sort of a screening throughout a year where he got fish from the various facilities, and in those analyses he could see that the gaping was not as high as that generally.
Yeah. So do you think that the problems of quality, flesh quality, are greater for fish than they would be for other food products like meat or vegetables and fruit? It's maybe not a very good question, but I just know that on television in the UK we are continually getting TV about quality of vegetables, and only the best ones go to the supermarkets, and there's such a lot of wastage and downgrading. I was wondering, you might not know this, but where do you think fish sit relative to other food products for waste and loss due to quality?
I think it's relatively high, because the fish has to be really fresh before people think it's, it doesn't take long before people think it's unappealing. These fillets or whatever it is, it has a sort of limited option to how to use it compared to maybe pork or whatever. There are more options in how to sort of maybe cover how bad it is, use it in other products and such. So I think it's the quality is relatively important for getting it sold.
Yeah, certainly when fish starts to go off, you know about it so maybe more so than some other food products. So moving on to it, more focus on the gaping. You mentioned as well that you get seasonal effects when it might be worse than other times of the year. You collected your samples for certainly your first 2 chapters, I think, in July. Was there a reason for collecting them in July? Is that the worst time of the year, or was that just the time of the year that you happened to be able to get the fish?
It wasn't exactly planned to be a certain period, but I know that I think the period late summer about there seems to be the worst season for it. I'm not sure why, but that is what we sort of, the information we got from the industry.
Okay. And whenever we'll come back onto this a little bit more when we look at the actual results chapters, but there's, from my reading, there is a sort of clear relationship between firmness and gaping. And I don't know which one happens first. Is firmness decreasing, then gaping occurs? You talk quite a lot about gaping, but you don't really mention very much about the firmness. Could you maybe tell me a little bit about what you actually think the firmness is, what actually is the underlying causes?
I think it can be sort of affected by some of the same things as gaping, and it can also co-occur, of course. But what is sort of seen in the industry, they say that the fillets can certainly be soft, but there's no gaping. And on the other hand, there can be gaping, and the fillet doesn't have this softness to it. So I don't think it's the exact same things that are going on, but maybe the same things affect it somehow. Some of the same things, at least. So, yeah. But I can't be quite sure, of course.
Yeah, so that brings us on a little bit to what actually, towards the end of your introduction, you start to talk about the different enzymes that may be responsible for breaking down this extracellular matrix. And you talk about the metallomatrix proteases, the family of those. And of course, we know these are important for remodeling. So the proteins which are present are clearly going to be important. And I was just wondering, did you ever come across the Danish researcher Flemming Jensen? He did quite a lot of proteomics on post-mortem quality.
Yeah.
And, I don't know if you know this at all. I haven't actually checked it, but I'm just wondering, with the proteins he identified, did you ever go and see were any of the things he found maybe might have been of interest to yourself?
Well, actually, I contacted Flemming Jensen, and following the mass spectrometry analysis, which were done in Boston, it was planned to also set up a similar analysis here in the Faroe Islands, and we worked quite a lot on that. But unfortunately, the instrument failed. And there was only one in the Faroe Islands. So we had to go to alternative solutions. And one of this was that I contacted Flemming Jensen about, he does these 2D gel analysis, and he can, and you can see the spots that are different, and put them through the mass spectrometry to identify them. So I sent him samples that I had extracted and such, but unfortunately, they didn't have high enough concentrations for him to do proper analysis on. But that is certainly something that I think is a very practical or applicable way of working with this issue.
That's really interesting. I had no idea you'd been in contact with him. I used to do, or still do, proteomics and a lot of gene expression work, and of course this type of work I felt lent itself to proteomics, because you want to look at the protein and what is present, not like the RNA level, but actually what is there 5, 6, 7 days post slaughter. So I find that really interesting. You also mentioned some enzymes called ADAMS, STADAMST. I can't remember what it is. Could you maybe remind me, if you can, what it actually was trying to degrade?
Let's see if I can find it.
It was near the end of the... It's on page 28.
Yeah.
At the bottom of page 28.
They act on proteoglycans, and unfortunately I don't know much about them. I just sort of, when reading up on these matrix metalloproteinases and sort of trying to sort of get other relevant also included, then these ADAMTs, or what they're called, seemed highly relevant, though I don't think it has been focused that much on them.
No, so anyway, just as part of my playing with your PhD, I went and had a look to see, did we find any of those genes in the salmon genome? And there was 2 copies of them, so but that's a little relevant section, which I'd never heard of these before, and I was quite interested to see. I think that's probably all the questions I have for the general introduction. I'll ask Philip. Yes, Philip's got his hand up.
Thanks, Sam. So I'll just start by saying congratulations on your very large and comprehensive thesis, and also your very clear and concise presentation today.
Thank you.
So I had a couple of questions for the introduction, and then of course I'll focus more on the microbiota stuff towards the latter stages, since that's kind of where my specialty lies. But so you had 3 papers where you talk about gut microbiota and salmon gut microbiome.
Yeah.
But I was kind of curious that in your introduction you don't really talk much about the gut microbiome in salmon. So and I know your main focus is on gaping as such, but I thought perhaps now we could have a quick discussion and how much do we know about the salmon gut microbiome? How big a role do you think it plays in host health, nutrition, the phenotype in general? I mean, we know that the gut microbiome has a huge impact in many animals, obviously us, humans, ruminants, herbivores in general. Fish is not something that is widely studied, so I'm kind of curious if you could kind of elaborate a bit there and let us know how much, what we know about the fish gut microbiome.
Well, there have been quite a few sort of fairly large studies on it by now. So the sort of the general overview of what it looks like, I think, is there, the basis. But the effect of it, and you know, what is good and what is bad and such, I think that is a bit more uncertain. But there have been quite a few studies also on, for example, food additives and such to sort of modify the microbiota in the intestines. But I don't think I've seen anything that is really implemented or sort of generally understood as this is really good and this works for improving this. So I think it's a bit in its early stages compared to other more us, for example, but also maybe more established farm animals and such.
Can I ask? This is a bad pun, but what is your gut feeling with this? Do you think that the gut microbiome has has a big role, a small role? What type of functions do you think it plays? Is it aiding in digestion of feed ingredients? Is it mainly transient? This is just a kind of what you think based on your experiences working with these samples in your thesis.
I think it seems to be quite species specific. So I think it has some function as to the immune defense and the mucosal barrier, and also what kind of food they forage, as to how it is graded down. I don't know what it's called. So yeah, I think it is important, but I think we don't know how important is it, and what exact functions does it have. But there are currently research on the way as well as to looking at for example, the mucosal in the intestines, and the sort of the interplay between the bacteria in there. What effect does it have on the immune system and such? So I think the next few years we will know a lot more about that. But generally, I think the microbiota in the intestines is important in any animal, I think.
Why do you think we see such large individual variation in the salmon gut? I mean, you see consistently large abundances of certain taxa, but then you see a lot of variation as well.
Yeah.
And that seems to just kind of put fishes apart from other kind of host microbiome ecosystems.
Oh, well, I don't really know why there is such large variation. It could be that some part of the microbiota is quite transient according to what they forage on, for example, or where specifically they are at. But while other parts of the microbiota is more consistent according to the species. And I don't think if that is more so in fish than in I don't know, other animals, it is.
Yeah, I mean, it's these are all questions that we all ask ourselves as well. So I'm just taking advantage of the fact that you've been studying this for three plus years, and maybe you have insights that could help the general kind of research community working on this. I think, yeah, I have more questions, intro related, but I can come back to those when we start talking about your later papers. So I'm happy to pass this back to Sam if you want to start moving through the actual articles.
Okay, thanks Ása for that discussion there. So Ása, we'll go to your first scientific paper, which, congratulations, published in Aquaculture Research. Gaping and loss of firmus and farm salmon closely correlated with post-slaughter cleaning and abdominal cavity. So I know that you had chosen to use these two parameters, cleaning and temperature, from the local input of your local aquaculture industry. You mentioned that, I think it was in the introduction. I'm just wondering, what was your input to this? Did they come to you or your research team and say, we want you to study this for a PhD, or was it the other way around? You wanted to look at gaping, and then you went and decided with them you would look at cleaning and temperature as your two main parameters, as opposed to season, feeding, stage, or whatever? So I'd just like to get a bit of a background to the development of the research plan.
Yeah, it was a bit of a combination, because we previously worked with this quality project where we looked at all these parameters, and the feedback that we got from the industry was that nobody really could get the fix on gaping. what is it and why is it and what do we do and such. So that it was sort of an interest that we got from hearing their sort of issues with it, and then also seeing the results from this former project, which sort of indicated some correlation between cleaning and gaping. And when we then talked to mainly the person mentioned in here, Dorita Nilsen, she said that this what they had experienced in their optimizations, it was that it was influenced by stress, which is sort of more agreed upon, but also by the temperature and the trials they had on cleaning it more meticulously. So therefore we sort of thought that try and set this up and try and get a closer look into what is it really and what happens, so that you know that when it's good, then you know why it's good, so that you can do something about it when it's not good.
Yeah. Okay. So you had I've got my questions written up here, but there's some things which come to my head very quickly. You used 30 fish, and we'll come back to that again. I mean, it was a small number, and in your discussion you say that certainly for chapter 2, the protocols, and you had a limitation for the number of samples you could put through some of the machines, and very clearly the biochemistry and the extraction process in the second results paper was very time limiting. But I didn't get the impression that there would have been such time limiting things for the first results chapter. So you used 30 fish, split them 2 groups of 15, and then you had 10 and 5. So actually some of your sample sizes were 5, which was quite low. You probably know that yourself. So I'm wondering at the experimental design stage, and I'm just wondering, did you ever think, or your team ever think about power analysis, where you can actually predict how many samples you need?
I think at that stage it was more of a question of what could we get at all. Because particular at that stage, the salmon farming industry in the Faroe Islands wasn't that sort of keen on doing these projects and research and involve in that. It isn't like in Norway, for example, where you have this that the industry puts a lot of money in these pools that researchers can sort of get money from and set up large experiments. Here you have to contact them sort of in person, and they have to be involved in this and agree upon how do we do this and everything. And at that time, it wasn't that usual to get 30 salmon. So
okay.
It is much better now, I am glad to say. But it's sort of it was sort of a development to sort of get the industry involved in research more, in projects and setting up these things along with us. So that's why it is a bit limited. But I know that well.
So that's a very honest answer.
Yeah,
I think you said quite.
But that's sort of how it was.
Okay, yeah. So one thing in your introduction and in the materials and methods here about how you scored gaping. You had a 0 to 3 scoring for, I suspect, a full fillet, and you showed a picture in your presentation about. And in your second paper, and I kept coming back to it because it's the same sample. Some of the samples you were able to have a good bit, a non-gaping bit, and a gaping section. Mm-hmm. So is gaping sort of, you get a localized zone of 5 or 10 centimeters of gaping. I know I've only ever seen pictures of gaping. I'll have to go back and look at salmon in the supermarket and see how many of them. But is this sort of localized and you'd always get it in one place, or could a salmon fillet have maybe 3 areas which have gaping and then good bits in between? I don't really get a feel for that.
Yeah, that was also sort of a problem in setting up the experiment, is that you don't have any idea what you're going to get and what you can sample and not. So you just have to see what's there and take what you can. And most of the time it's not sort of gaping all over the place. It's just that when it is a bit severe gaping and you want to have an intact sample, you sort of, I was sort of reluctant to sort of take it, you know, where there's one connecting tissue alongside with a bunch of holes, because I wanted to be sure that the molecular structure was clearly different from where it was gaping. So that's why it was I was a bit conservative, that I wanted it to be a region of, you know, fully intact, or a region with gaping. And that's why I couldn't get both intact and gaping from, you know, more of them.
Okay, that's fine. Yeah. So you used for your general quality, you used 2 methods, as I could work out. You used something called QIM, Quality Index Method, and the Norwegian Quality Cut Method. So the Quality Index Method, was that something your lab had set up on its? Is that coming from your research group?
No,
and the Norwegian one is from Norway, or are they both?
Yeah, it is the one established previously, and which was sort of, I think it was a Icelandic Sveinsdottir or something.
Okay.
Yeah, I can't remember. Yeah, who sort of implemented it in salmon, and so it's not something that we invented. We just implemented it from that in this previous project. So it's a sort of I don't know, international standard, but I don't know how far it reaches outside the Nordic countries.
I don't know.
Yeah.
Okay, right. So that's sort of questions about your methods and the materials you use for that chapter. We'll move on to the results now and discussion about what you actually found out. So you had fish which have either been cleaned and not cleaned and kept for 3 hours at an ambient temperature or a low temperature, and it was quite clear, we think, that period of 3 hours of the ambient temperature did not really have an impact. Is that correct?
Yeah. From these results, it sort of it didn't show to have an impact, but I think we have to be careful of assuming that, because of this failure in the cooling system, and because the average temperature was about 4.6 or something, when they usually keep it more like 2 or something. So there might be a difference between keeping the ice really chilled at 2 degrees and when it's 4 or 5 degrees, which has more of a relevance than if it's from 5 degrees to 11.
Yep. So we already discussed your sampling and why you had 30 fish, and you've explained that it was difficult to get fish at that time, because one of my comments here was, when your cooling machine didn't work, why did you not just start again? Well,
Yeah.
you know, because
I
as soon as you know, if it was difficult to get samples, I understand, and therefore you were able to disregard the temperature effect.
Yeah, yeah.
Yeah, it's a pity that you probably lost, you know, quite a bit of data there.
Yeah.
Or information. On figure 3 in this paper, yeah, you have a relationship between remains in abdominal cavity, a normalized log number versus gaping score, and then below the remains in the abdominal cavity against firmness score, and they show a very good relationship. But I could not find any information about how you scored the amount of remains in the abdominal cavity. How you, on the x-axis of those graphs, what those numbers refer to?
Yeah, it was scored also 0 to 3, and that was something that it is a subjective score because it's not automized by photography or anything. So it is training of staff to establish these sort of groupings, and that was also established in this former project that I mentioned, so that it was only Magnus Paul Magnussen who always performed these estimates on all the salmon. So it was based on his methods, how it was graded exactly.
Okay.
I had something in the presentation. There was a sort of a bit of a description of that, as the same with gaping score.
I just whenever I was reading it, I couldn't work out.
But it's not in the paper. No, true. No. That's true.
It's okay. And you sort of hypothesize that that it's to do with the cleaning that affects the gaping and not temperature, which would suggest that there's enzymes or something that's come through from the cleaning, which is starting to degrade the extracellular matrix. So there's various, and you have quite a lot of discussion in this chapter, which leads nicely on to the next chapter. So there's various options for the enzymes responsible for degradation of the extracellular matrix. You've got these metalloproteases, you've got lysosomes, but other bacterial collagenases. And of course, you decided that you would probably go down the bacterial collagenase route rather than the fish sourced endogenous enzymes, and you make justification for it nicely in the next chapter. But at this stage, I'm just thinking about the timeline of your PhD. Whenever you had done this chapter, were you at that stage, were you thinking about microbiota, or did microbiota ideas come after the second chapter? Or after the second paper?
I think it came sort of as it was a bit of both. It was a thought that we had when we sort of thought of there's something of the remains, then there certainly is a potential for that it is from salmon, of course, but there's also this potential that it's from the bacteria, and also that it's a combination that it sort of interacts somehow. So that's why we sort of had this potential in mind, but we didn't have much opportunities at the time to do sequencing. So it took a bit of a while before we could sort of go on to that. Okay, well, thanks very much. I'm going to finish that chapter. Is there anything Philip wants to say about that chapter, or we keep going?
It's okay. I'll wait till.
That's fine. Okay, we'll move on to your second published paper, characterization of glycoaminoglycans and gaping in connective tissue of farmed salmon, Philip's by mass spectrometry. So, Ása, were you a biochemist in a previous life, or did you learn all the biochemistry during your PhD?
I basically know what I've written, and that's it.
Okay, yeah. So I did biochemistry a little bit as an undergraduate, but so this is not my area of expertise. I can understand the bigger picture of it, and it's been really interesting to read with a good focus on something I'm not particularly, an expert on. So I did find it very interesting, and again leads to your third results chapter. So anyway, you're talking about these GAGs, these glycosaminoglycans, I'll not be able to say that very much. So you actually decided to look at the amount of these and the diversity of these molecules as opposed to enzyme activities, coming back to the enzymes. So it was clearly at the decision point, you could have tried to look at the GAGs or you could have tried to look at the enzyme activity. And I wonder where that decision, how that decision was made.
We wanted to look at the connective tissue itself. That was fairly early in the process that we wanted to do that. From the literature, we're trying to sort of first get an idea of what could it be in the connective tissue. What are the potentials to look at? And then we came down to these glycosaminoglycans. And we Jonhard contacted Josiah, which is somewhat of an expert on this area. So that was a great opportunity. But the thing with the enzymes, I think that is the interesting area to move into. Because but I think the sort of the main problems with looking at the enzymes is how do you sample your enzymes without interfering with the fish and knowing if it induces gaping. So how do you do this timeline of what happens without and being able to say that it's not because you moved it around or did this and that, that's why it is gaping, or that's not why it's not gaping, because it doesn't always appear straight away. Sometimes it takes several days before the gaping appears. And how do you know where to sample? And so I think how to set up such a project that you can sort of connect the enzymes with what happens. I think that is sort of the main issue, I think. But otherwise, the enzymes is a very logical place to go to study it further.
Yeah, I read through the extraction methodology. It seemed like quite laborious and time-consuming. Only 3 days, and you give a nice flow diagram in your appendix about the actual protocol. So, did it take quite a long time to get this going in your lab? Was a lot of trial and error, or did you just go in and you
because this was done in the States at the lab where Josiah has his group. So they had sort of established this process already. So I was just introduced and taught this process, and then I could sort of proceed with the samples myself. So it's completely sort of their setup.
So did you get an opportunity to travel to do this in the States?
Yeah, I was there for 3 months to do this.
Right. Very good. And you said that there was some differences between your method and a method of She, which was a paper. I didn't read the paper in 2007. I was just wondering, because you were doing fish, did you have to change the protocol quite a lot?
You have to see what you are referring to.
It was just a technical question. I mean, it was really, was there a lot of changes you had to do to try and get the protocol to work for salmon flesh?
No, I don't think there were a lot of issues. The only issue was that it had a lot of noise in the total ion chromatogram, and we tried with this alcohol, chilled alcohol, but it didn't change how it appeared. So we tried it without, and it worked fine. Otherwise, the setup was the same.
Okay, and you mentioned it in your presentation as well that you had to do some pooling of samples.
Yeah,
it was probably quite disappointing for you. What was the actual reason? Was it because you only had a certain number of mass spec runs you were allowed to do, Was it a cost thing, or was it where exactly was it? It's hard to say.
Yeah, it's completely different from the problem we have here. In the lab where Josiah was and other professors had their group, there were several types of these mass spectrometries, and for each mass spectrometry there was a gatekeeper, which is the person who sort of has to decide who runs now and helps them set it up and fixes all the problems with it. But these machines are tightly booked all the time. There are lots and lots of students there, all pressing for access. So when I had these 43 samples, they just said, you have to reduce that. Yeah, so and sort of I tried to sort of get it a bit down, but they weren't happy until it turned out to be 32.
Okay,
so
right,
yeah,
that's so it's out of your hands, really. But
yeah.
Do you think that impacted on your variance in your results, and do you think it impacted your statistical analysis at the end of the?
Yeah,
because putting samples reduced variance for that group.
Yeah, it made it more difficult to find any significant results, so it reduced that possibility a bit. So if we had run the 43 samples, we might have been able to see something more, maybe. And it can also be seen in the table where we compare all the samples, that the same amount of monosulfated disaccharide in chondroitin sulfate. It is not significant, but it's fairly low.
Almost,
so it's almost there. So maybe that would have sort of made an impact. But I can only speculate on that. But it reduced the chances of finding significant results.
Yep. Okay, so we'll move on to results now, and you'll start off with the chondroitin sulfate, because that's where you obviously find your most interesting results. And you did your mass spec, and then you were able to close in and get your peaks and your abundances, which was very nice. So a technical question is, you had unknown ion species were assigned 10101.
Yeah.
Why did you decide to assign the unknown ones to that? Was it because that was the most abundant, or because, I just don't know.
You mean that the chondroitin sulfate was this, or
and heparan
sulfate not?
Table 1.
Yeah, table one. Yeah.
And your footnotes, footnote B, ions with unknown function assigned as 10101.
Unknown modification assigned. Yeah.
Why did you just choose to call it that as opposed to something else?
This was also the peaks that I looked at, because I couldn't run MS, so I couldn't sort of guarantee that the peaks that I were looking at were exactly the compounds that that are on the table. So this was all established also beforehand by their previous work, so that you basically got this sort of schedule or table that you could fill out and look at all the MZ values and sort of extract this and that. And this, with the unknown modification that they had assigned, is what they had done. And this was also mentioned when I submitted the paper by the reviewers, that, how do you know it is this? And I could only refer the question to Josiah, and he only responded with saying that it was assigned based on retention time.
Okay.
It was extracted as and digested as chondroitin sulfate, and the retention time was what they based it on. So I'm not sure they've done MS on it, but I couldn't do it myself to check. So
Okay. It wasn't a large part for large discussion. It was just again a small comment I had.
Yep.
So you find no difference between gaping and intact fish when you looked at individual fish, but when you looked at these fish which had regions of gaping and intact, you did then pick up difference in the sulfate level, the sulfation level of the CS, chondroitin sulfate, and particularly for the 10111. So this, and on table 3, you had the p-value of p is less than 0.0 or equals 0.027 to say that 1011 was significantly different. And then at the bottom half of that table, you've got the percentage distribution for the other disaccharides for the CS.
Yeah.
Can you just? I was a little bit confused about this bottom section of the table and how it related to that top bit.
Yeah, I think the sort of relative the relative content of this non-sulfated and monosulfated and desulfated is that there are these 3 types, and you see how much is there of relatively, while when you look at the abundance, it appears to be a much higher variation between salmon. So I think it comes from that that you can't see the significance in the abundances because of the variation in there, but you see it in the relative distribution between the types internally. So I think that is sort of how that plays out.
Yep. And the next figure I wanted to take a look at was figure 3, where you had the comparison of the 3 CS disaccharide compositions.
Yeah.
And so this is probably one of the key results from your paper, which again leads on to trying to work out what's happening. So what we're seeing generally is a decrease in the sulfation, but during gaping, but an increase in the monosulfated molecules. Is that correct?
So gaping has an increased amount of non-sulfated and
so there's desulfation of them,
and lower amounts of the sulfated, both of the sulfated types. So it somehow it seems that it has lost some of its sulfate groups comparative to the intact ones.
Yeah. So this then, of course, leads you to your probably conclusion that it's got to have endosulfatases that knock off the sulfate molecules.
Yeah, as a possibility.
Yeah. And of course, the sulfate molecules are probably used for the structure and integrity of the extracellular matrix, or how it binds together the collagen molecules, and whenever these sulfate molecules get removed, I'm suspecting that's what causes the solubility of the collagen.
Yeah.
I think. So then you go on, you talk about where these enzymes might come from, and your conclusion is that there are none of these enzymes in eukaryotic genomes, which is why you think they're microbial.
Oh, genomes.
You said no eukaryotic
currently
CS
no.
endosulfases have yet been reported.
Yeah, I couldn't find any reports on that. I don't know if you can correct me, maybe.
No, I can't correct you. I just had a very quick look this morning to see just the only ones I could find, like you mentioned, were the endosulfases. There was a couple of proteins which are well characterized for the heparin sulfate, but I ultimately, I didn't do an extension. I just did a text search, to be honest. But yeah, and this gave you the potential that if this reaction was happening, it must have been from, or potentially could have been from microbial contamination and the enzymes coming from them.
Yeah.
So that was interesting. The heparin sulfate, there was a lot of talk about the CS anymore. I don't think I've got any more to look at about those, but yeah, you didn't find any differences in the heparin. There was no clear responses, but you did find one factor which was found to be different, which was to do with the degree of sulfation. In table 4, there was something that was so marginally significant.
Yeah.
Is that what you were mentioning about maybe your replicate numbers, etc.? It might have.
Again, what did you say about replica?
Well, you in most of your paper, you're saying there was no difference found for the heparin sulfate, but actually you do find one thing when you paired t-test, which was different but not found different in the Welch's t-test.
Yeah. And I don't have any sort of theory or of why this is or what it could mean or anything. So so I couldn't elaborate much on that, unfortunately.
Right. Okay. So I've got another sort of question for about one of your, well, two of the graphs actually. Figure 5. Look at figure 5. On your x-axis, you have things like DO, AO, and D2AO. There was nowhere in the paper that described what these were. At least I couldn't.
No, that's correct. Yeah.
And it's doing acetylation, I believe.
Mm-hmm. This is because this is what the nomenclature, or what's it called, that they used in that lab, and this is sort of the commonly used language for it. So I didn't know that either. I just applied it as well, and my thought was that the people who read these kind of papers, they know what this means.
Okay, so maybe I should have gone back and looked at the publication 63, which probably explained that. But for someone lazy like me, it probably would have been better to have just a little table explaining what the nomenclature was.
Yeah, absolutely.
But anyway, I sort of was able to work it out, but I never went. I didn't go and read the paper. Yeah. So the last graph I'd like to ask you about was figure 6, where you have a relationship between the heparin sulfate chain length and your NRE, which was
non-reducing end.
Yeah, non-reducing end. So there does look like there's a difference in the relationship there between certainly the top line, which is the U2OA, which is steeper in the intact than it is in the gaping. Did you or anyone try and do any kind of further statistical analysis on that to see if you could maybe like modeling or some kind of data analysis? Put anything out from that?
No, I didn't try because the sort of values are fairly similar, but it's more the sort of that it follows a line in the intact tissue much better than it does in the gaping tissue. So we sort of left it at this stage, where it was only compared internally.
Okay, right. We've had a fairly thorough discussion of that, and I see now that's almost exactly an hour I've been talking to you. So I'm going to, thanks very much, Asa, and I might chip in, or I'll certainly chip in later on at the very, very end anyway, if I don't ask some additional questions from Philip. But I think I'll hand back over to Philip now and let him talk about the microbiota. Okay, thanks very much.
Yep.
Thank you.
Okay, thanks. Yeah, I'm hopefully not going to go for 1 hour, but we'll see how it goes. So, yeah, I'd like to start by just a more general kind of question. So, in terms of working with salmon gut microbiota, there's a lot of technical challenges to overcome.
Yeah.
And I assume that you've come across many of those yourself in terms of low biomass, host contamination. So just first of all, just from your own experiences and your own troubleshooting, some ideas you have to overcome these technical challenges.
Yeah. Well, you have to be really careful at every stage. So all the way from the sampling, it has to be very consistent. A few people working on it, and sort of to reduce any chance of inducing something or introducing contamination. And usually we do negative tests with the extraction that we sort of test afterwards, and in the PCR, the initial PCR. But we and sometimes we get sort of signals from that, but we haven't tried to sequence any of it.
You didn't sequence any of your negative controls?
No, we haven't sequenced negative controls, so I haven't tried that. But I like the idea of that some papers suggest that sequencing this negative controls, and if they do show some reads, and to sort of publish it or show it along with the other samples, and not to remove samples, because that was sort of the reason why I'm not so keen on removing bacteria, because they are, typical soil or environmental bacteria that sometimes are known to come from lab work or extraction kits or something, because we work with environmental samples. So it's not as we work on lung tissue or something that is not supposed to have these things. If you work on storage eyes and things like that, you sort of expect to have potentially bacteria that are known to somehow contaminate the samples. So that's why we haven't proceeded to sequence it. But I think presenting it along with the results would sort of give an opportunity to discuss that potential failure.
Then how would you kind of separate potential environmental kind of origins for taxa or potential contaminants that are not necessarily associated with ice or rinsing water? I mean, for example, coming from yourself, or even kit reagents. So I mean, kit reagents are known to in instances give a PCR product and sequences if you sequence that product.
Yeah. And how do you? What was the question?
So how would you kind of identify which source of contamination?
If I sequence that, you mean?
If you don't sequence it, if you don't sequence your negative controls, how can you kind of place an assignment of where this contamination is coming from?
You cannot you cannot be sure if you do the sequencing and you don't have these sequence controls, the negative controls. You can't be sure that there haven't been introduced contaminations. So there were only negative controls for example, in the extraction kits that we sort of, if you see the DNA concentration or signals from from any of that, then you know there is something going on, but not in the sequencing itself. And then we can't be quite sure that nothing is there that shouldn't be.
Did you also include your rinsing water as a sample to amplify and sequence?
Not the water itself that we rinsed it with, but the storage ice was used. Yeah.
Do you think perhaps the rinsing water may have an effect on what you see from your mucosal, like your mucus layer on the skin?
Yeah, it could of course have had an effect there. Yeah, absolutely.
Yeah.
Yeah.
Okay. So if we go back to this technical issues, the low biomass is obviously a big challenge working with these samples. You can see when you listed your OTUs, your samples and the amount of sequences that you had a lot of variation. So huge variation of recovered 16S sequences that are coming from each one. Did you normalize DNA concentrations before you applied this to your MySeq, or do you think there's other underlying factors that cause this kind of variation in your recovered sequence counts?
A bit of both, perhaps, because some just have a bit of a lower concentration, so you can't add the amount that you would like to. So that sort of contributes as one part. And also in the other parts, because you're not sure how the other factors affect it further down along the line. But mainly, if we had problems in the normalization, it was because the concentrations were too low. So they had to be just analyzed in that way.
Okay, so you didn't have enough PCR product to increase it, so you would have a normalized distribution across your pooled samples.
Yeah.
What was the PCR cycles? How many PCR cycles did you run?
26, I think, on the first.
Yeah, and so you didn't increase. You capped it at 26 across all.
Yeah.
That's actually quite good. That would actually decrease your chances of getting contamination from hosts from your kits, for example.
Yeah. No.
So when you talk about your results here, you compare them against other studies, but I'm kind of curious as to the methods on how you compare what you saw from your data against what others have seen from their data. So what kind of methods did you use to do that? So I've got a quote here from your discussion. In general, so you talk about the bacteria presented at high abundance in the skin did not correlate well with previous studies.
Yeah. I saw that, and I think that is sort of a bad description or a phrase.
Okay.
I think
that's okay. Yeah.
But
if you were going to do that, for example, okay.
Yeah.
How would you do that?
Compare.
Yeah, if you wanted to do a analysis to support that statement.
Well, I mean, you have to firstly look at the literature and also see what kind of methods have been used to see if they could be, if the comparisons can be made properly. But yeah, that's about it, I think. Because I think that the main thing with this, that it didn't look that similar, was more that I thought that the skin mucus looked like it had other variables in, like from the rinsing water and such. And when you look at how the other had sampled their skin mucus, it wasn't in this kind of setting where the salmon had been through a processing part and in storage water and all this. So it hadn't in other papers, it hadn't been influenced by the things that we had put it through.
Okay. Is it possible to put your slides back up again? Because I'd like to talk to a couple of slides that you had.
Yeah, I think the technician will.
So if we go to slide 30. Okay. Okay, so here you talked about diversity.
Yeah.
You're talking about different levels of the diversity. But would you say that using visual kind of examination of bar charts is a way to kind of estimate and compare diversity between different samples?
No, not visually. Are they generally more diverse? Is that what you mean? The comment?
Yeah, so what kind of methods, what kind of approaches would you use if you wanted to assess the diversity between two different samples?
That would be
How should you do
by cow1 or number of OTUs or any of these alpha diversity estimates. Okay. So that is. I think this comment here is sort of, yeah, it's sort of a bit more, as you can see, not
sort
sure. Yes, it's not the sort of statistical test.
Usually the reviewer, the examiner number 2, my job is to pick you up a bit on technical stuff. So, but I understand that when you're presenting this, obviously you can't go into that type of detail. But if we can go to the next slide. So I'm curious here. So one of the things I got excited about when I read your papers is that you had a look at the different sections of the gut, because this is something that's not usually done. Mm-hmm. So like, for instance, the pyloric ceca and the midsection. So looking at how they compare to the hindgut, because usually we always assume that the hindgut is where the bulk of fermentation occurs.
The bulk
so,
of
but
what?
I'm curious here why you pooled all your intestinal regions in your PCA plot.
Oh yeah, I think they are so concentrated on one that it's hard to differentiate them. So
Okay,
so you saw the same taxa basically dominating across the pyloric ceca, the midsection.
Yeah, it was very similar, and I think I compared the internally the pyloric ceca and the mid-intestine and distal intestine, and didn't show any significant differences between them, because there's a relatively, again, as you mentioned, a relatively large variation in the individual variation. So I haven't separated them in that plot either, and also because they are so tightly grouped in there that.
If you were to remove, say for example, if you reran your PCA and you just included your gastrointestinal samples, so your midsection, your pyloric ceca, your hindgut, would you expect to see them also grouping as tightly as they are here, or did you see?
I think there would be a bit of a crisscross between them. I don't think they would group all that tightly into different sections, no. As I can recall from the individual differences.
Okay, so while we're here,
but maybe
if we can go to the next slide.
Yeah.
Yeah, so you saw box 3.
Yeah.
An outlier, and you also saw in terms of the activity of your gelatinase. There was an outlier, much more activity.
Yeah.
So I guess this leads into a hypothesis here that do you think it's this taxa that is extremely high abundant is connecting to this activity that you see?
That I don't know. It could be that the mycoplasma or mycoplasma could be involved in this. But you can't be certain. It could also be just an indicator, because when you have this intestinal material transferred, you also have the bacteria. So that's the part where you can't distinguish between that yet. the enzymes,
Sure.
where they come from.
Yeah, so the thing is here, you're using 16S to identify your taxa.
Yeah.
How would you say, okay, so this is more of a hypothetical question. This is beyond what you've done, but say if we said, okay, you've got unlimited budget, unlimited fish, what are the experiments that you do to try and answer this question? How would you go and kind of and try and attribute this taxa to this activity?
Well, you'd have to see if maybe this whole genome sequencing, for example, so that you can see what potential the bacteria have for this activity.
Yeah. So say you sequence genomes from all your samples and you annotate these genomes, you get a sense, but that's still not going to connect the actual activity, is it? I mean, it's going to tell you its potential.
Yeah.
But it's not going to tell you if it's actually contributing to that phenotype that you're seeing.
Yeah, you have to have gene expression as well. Yeah.
Okay, so I won't focus too much on the last two papers because, as I can see from your presentation, they're a little bit of an outlier in a sense for your thesis. But there were some points and some questions that came from them. More curiosity, actually.
Yeah.
So this, for one, is you use a completely different set of primers.
Yeah.
And I was kind of curious as to the timeline. I'm guessing this is your initial pilot study before you got these samples and started working with MySeq.
Yeah. This initial pilot study has a lot of flaws, but this was our first attempt to work with next generation sequencing. So none of us had any experience with it, and no sequencing machine was in the country. So this was our attempt to sort of just set out and try to see if we could see anything interesting, if this would be a line of inquiry that would be, interesting. So all of us working on it were inexperienced in this, and therefore I think that many of the technical things aren't that great. But it has some features that are interesting, so that is why it's included, and also it's important in the process of getting to the study that has been published. And it was also instrumental in getting a sequencing instrument to the country, as it was one of the studies that sort of showed that we had interest in working with these things. So it's sort of a part of a process in getting to where we can work properly with such methods.
Do you know why you didn't see any results for the Bacteroidetes, the phylum Bacteroidetes?
It was the primer that didn't that wasn't selected, wasn't checked to cover that.
Okay.
Yeah.
And so your qPCR, this is Bacteroides specific primers.
Yeah.
Yeah. So how many? I'm curious because you didn't really talk much about this data in this manuscript. You mentioned that it didn't work, and they used qPCR to estimate cell numbers. Did you see high levels, moderate levels, low levels of the Bacteroidetes? Because I couldn't find actual numbers to see how much there was.
Okay. Yeah. Fairly high numbers, I think. So it was, moderate to high, if I can remember correctly.
Okay.
Yeah.
This might be a little bit off topic, and it's okay if you don't know, but the Bacteroidetes, do you know much about what the normal associated kind of phenotypes are?
No, not offhand, but I think they have they have a lot of potential for this sort of enzyme activity, as I can remember. Is that true? The collagenase?
Yeah, that's true. Yeah. So, and one of the interesting things here, this is something that's come that I actually I missed when I read your thesis the first time, but it's come through today. It's picked up on me. Is the fact the sulfatases. Yeah. So at least in the gut environment, not in fish because I haven't looked there yet, but in other systems they encode a high number of sulfatases.
Okay.
The Bacteroidetes.
Yeah. Yeah.
So they're often involved in polysaccharide degradation from plants, but also from host-associated glycans.
Okay.
So this is something that's basically light bulb when I was listening to your presentation earlier today. Okay, so I'll think I've got a couple more. So you just quickly touched on this in your presentation, and then this is in your last manuscript, your third microbiota one. Yep. You talked about methods and pipelines and approaches. So I'm kind of interested here where you looked at comparing different pipelines.
Why?
And that is curious. It was nice that you did do this, but I'm curious as to why you didn't have kind of controls set up to help you do this, especially when you looked at databases. So for one method, you had one database, and for the other method, you had another database. Yeah. So I'm kind of wondering if you think this have influence on your results, and is this really the right way?
Yeah, as it is now, it is sort of impossible to know exactly which parameter has the most effect on this and that. But this is still a work in progress, and the next thing is to I've moved on to using QIIME 2, and the idea is to analyze the same data in QIIME 2 using the same pipeline, but at the finish use both green genes and Silva, and then you have a comparison of just the databases.
Do you know why green genes was largely inferior to the Silva database?
Well, it hasn't been updated for a long time, and it is kind of surprising, but it's still being used quite a lot, and some argue that it's at some points it is better than Silva or other, but as long as if you trust Silva to correctly ascertain the bacteria, then I think it is superior as it has shown.
Well, the thing is, green genes has actually been discontinued in many ways. So the authors that developed it don't do it anymore, and they actually don't use it anymore. No. So I would definitely advise against not using green genes for this type of work. Okay, so when you've used 16S, you have a nice list of observations from your work, and I mean the fact that you were able to get recovery. I mean, you only had a small number of samples, but you were able to get high recovery with low PCR cycles. So I'm actually kind of jealous of that because a lot of times we work with this, we have a lot of issues even at this stage. The hypothesis that you get from this, I mean, the thing is 16S is really difficult to get solid answers. So this is more of a kind of ending kind of future perspectives type question. But what are the main hypotheses that you think that you've developed from this data? So you have your 16S data, your list of varying abundances. You have all this phenotypic data from your work with the fish. What are the main hypotheses you have? Is the first question I have. And then this goes on to, this is the end of your PhD, we're celebrating the finish. But as a scientist, you must have curiosity to kind of follow some of these things. So if you had more money, more time, what are the ways you would go about trying to answer these new hypotheses that have come from this analysis?
I think it's sort of this endosulfatase idea is sort of interesting, though it's sort of it is in some ways a bit of a stretch, because it's sort of hypothetical, part of the way. But that would be interesting to see if that is correct. That is what is causing the changes in the glycosaminoglycans. But it hasn't been established if these changes in the glycosaminoglycans, if that is affected, or if that is part of the gaping problem, or it's just it's because of the gaping that it happens, not the cause to gaping. So there are a lot of holes in this sort of chain of reasoning that you have to sort of look at. But I would look more closely at the enzymes themselves, enzyme activity, and sort of what kind of enzymes are active here. Are they bacterial, or are they from the salmon? And how do they affect connective tissue, for example, glycosaminoglycans?
I guess this comes back to, and on this topic, the localization of these enzymes. So you have your gastrointestinal region, and then these enzymes have to get from there to the tissue. Do you think this is a contamination issue, or is there some kind of transition of or movement? I mean, how are these enzymes getting, say, from the bacteria in the gut to the tissue?
Yeah, I think it's during the processing that it is not cleaned away. So it's there when it's gutted, and the whole comes into the intestine, and it sort of spills out. So I think it's during the processing, and it's not being completely removed. So I think that's where it comes from. Yeah.
Yeah. So I guess one of the things, and this is something that's come from today for me, from listening to yourself and to Sam, is when we talked about proteomics on the tissue. I would argue that you should also be doing proteomics on the gut, because you'll get identification of bacterial enzymes as well, and also you could use this for localization.
Yeah. Yes.
Yeah. Yeah. I mean, I have to say, I'm not going to talk for too much longer, because it's obvious to me that you have a good handle on what you've done, interpreting the results that you've gotten. So I'm happy here. Sam, if you have anything else you'd like to add to these three papers?
I've got one very small question, Ása.
Yeah.
It's related to your paper where you compare the microbiota across 4 different species.
Yeah.
3 of the species were wild caught and 1 was farmed. The wild caught fish are probably all carnivorous fish. And certainly in your digester, we know that from microbiota, a lot of what you actually are, your microbiota is actually as a result of the feedstuffs. Did you see any signature at all of vegetable material from the feeding of probably there's always plant-derived product in the salmon feeds nowadays. Did you see any signature of that at all?
No, I don't think so.
Okay, no, I was just wondering because I'm always concerned doing microbiota in the gut. Am I looking at commensals, attached bacteria, or just a transient bacteria? It's always a worry for me, is all I've been doing. It's a good thing for food. I don't know. And I was just wondering because the 3 wild fish may have had very similar diets, and the salmon, I think, might have had a different diet.
Yeah, so the salmon has eats these pellets, and there's more and more pressure on to not use marine-derived material in the food for salmon. but it has a lot of side effects if you use too much plant material. So the feed is produced that they use is produced here in the Faroe Islands and has quite a high marine part in it. So it's not as high in plant material as abroad.
Yeah, I've enjoyed reading your chapters. I'm not going to ask any more. Philip's actually asked some very focused questions, and I'm happy with those 3 chapters. So I've nothing else to add there, really.
Thank you.
So I don't know. As I said, it's obvious to me that that you know exactly what you've done and have good handle on not just the methods but also the challenges and technical issues that go with them. So for me personally, I don't have any kind of more grilling type questions, that's for sure. So I'm happy to, at least from my side, to let you relax.
Thank you.
Yeah, thank you. We thank the opponents for the questions. Now there are questions ex auditorio. I haven't gotten any emails with questions, and there are no questions yet in the Zoom Q&A. I don't know if there are any questions here in the room. Then I think we will go on. So now there will be a break while the committee withdraws to assess the defense. When they reach a conclusion, they will come back here on Zoom and YouTube. So we'll take a break and they will leave us now. And we'll be back in a few minutes.
Hello, can you hear me?
Yes.
There is a little problem. The technicians have disappeared.
So, welcome back. I will now give the word to the chair of the committee, Sissel.
Yes, you hear me here? So I've had the honor to chair this committee, and it's been I really enjoyed reading this thesis and also listened to this defense today. So in this committee we had discussions about the thesis and also the defense, and we all agree that you did a very good job also. I can't see you here, but and we would like to congratulate you with a very good job. So you're now a doctor, so congratulations.
And then I'll also congratulate you with a diploma.
Oh, thank you.
And some flowers.
Thank you very much.
And we'll have to wait with shaking hands.
Yes.
Also, I want to thank everybody that has been involved in the defense, the committee, the advisors, technicians, and thank to Ása for the presentation.
Yes.
Thank you, everybody. I think we'll give them all a hand.
Yes, thank you very much for the time and effort you put into this. Thank you.
And this concludes the session, I think. Thank you.
So we leave, or is this finished?
Yeah, well, now we stop the streaming on YouTube. If you want to say something more, you're welcome. But this is the part of the formal thing. Did you send me the email as well with signatures, or?
Sir, I hear you a little bit, not so good, but we will send you the signatures in a minute or so.
Yeah, fine.
Yeah. Okay. We leave then. Thank you, and have a good celebration. We wish we could be together with you today, but
yeah.
Congratulations, Ása.
Thank you very
congratulations.
much.
Bye then.
Goodbye.
Thanks, Philip.
Yeah, thank you. Thanks.
Thank you.
Thanks.
Thank you. Bye.
Thank you.
Bye.